As you may guess, this is Pittsburgh, and I am somewhere back here among these trees talking to you at this moment. What I'm going to talk to you about is the basal ganglia. Okay, now I recognize that some of you in the audience are very interested in basal ganglia, while others may not be and may know little about it. So the way I tried to structure this talk is it's going to have three parts in order of decreasing length. And in each part, there will be a focus on the basal ganglia, but there will also be some methodology that I think is of general interest to anyone studying neuroscience, at least from a computational perspective. So I hope everyone will find something of interest in the talk. So in the first part, I'm going to talk about some possible roles of the basal ganglia in choosing actions, as well as tuning how we act. And to do that, I'm going to introduce a technique that we call an upward mapping that allows us to connect parameters of a neural system to behavioral features, mathematically / statistically. Okay, in the second part of the talk, I'm going to focus on some surprising data about some responses in the basal ganglia to inputs from upstream. These relate to synaptic inhibition. Okay, so even if you're not interested in basal ganglia, many of us think about synaptic inhibition, and what I'm going to say hopefully has implications for how we think about and model synaptic inhibition. Okay, and then in the third short end part of the talk, I'm going to talk about some maybe unexpected delta band oscillations that we find in the basal ganglia, where they may come from. And that's a low frequency band. And so in general, there are difficulties with identifying real power at low frequency because of the sort of 1/f noise spectrum that can contaminate low frequencies. And so I'm going to introduce a simple but effective method that we use to deal with that problem. Okay. All right. So to orient everybody, let's start with this simple question of what are the basal ganglia? And this is an easy one because the name is completely descriptive. Okay, they are basal ganglia, right? So they're down in the base of the brain, and they're collections of neurons in these different subpopulations. So in this diagram with the coronal section, we see, for example, the caudate, putamen, globus pallidus, right? These regions deep down in the brain are part of the basal ganglia. You can see where they're located relative to brainstem and so on. And in this slice over here, this cartoon, we see also, again, the caudate and putamen, globus pallidus, and some other regions, substantia nigra and subthalamic nucleus, that are also basal ganglia regions. Okay, you don't have to memorize the names. Okay, I'll give you that, but this is where they are and what they look like roughly in terms of where they are in the brain. Okay, so then why are they interesting? So one of the factors that makes the basal ganglia interesting is they're present throughout all sorts of different animal species. Okay, all the way down to the lamprey, even, as Grillner and his collaborators have discussed. So this suggests that they may be performing some sort of really fundamental functions that are important to all animals. Now, it might be a boring function, like some of the other automatic functions, but nonetheless, this suggests they may be important and interesting. Another reason the basal ganglia come up a lot is because they're very important in disease, specifically Parkinson's disease. So to help engage everyone here, I have a little simple quiz for you, right? I have two brain samples here from a part of the basal ganglia, the substantia nigra. And so you can look at these two and try to decide, which one of these is from control conditions and which is from Parkinsonian conditions. Okay, and so you might be tempted to think, this sample here is the Parkinsonian sample, right? Because it has these sort of black bands, right? That looks like maybe a mark of disease or something. But in fact, this is the substantia nigra, right? So it's supposed to have black stuff. And so what happens in Parkinsonism is that the cells in this region actually die off. And so in the Parkinsonian case, this black stuff is gone. And this leads in various ways that are not so well understood to a variety of changes in activity in the basal ganglia and to the symptoms of Parkinson's disease. Okay, so that becomes a reason to study the basal ganglia. Okay, so we have the functional side and the dysfunctional side. Okay, now coming back to the functional side, for an area that is conserved throughout so many species, function is surprisingly elusive. So there are frameworks dating back to the 80s, after the basic anatomy and pathways had been worked out, that pose a potential way that the basal ganglia could function. Okay, and this is sort of this action gating model. Okay, so here we see various basal ganglia regions in the black dashed zone, getting input from cortex, giving output to the thalamus. And you can see some of these pathways are inhibitory, some are excitatory. So the principle here of this framework is that the thalamic excitatory signal is needed to allow action to proceed. Okay, and the basal ganglia inhibitory output then acts as a brake on this action signal, preventing actions from going ahead. So you have to remove the brake to act. Okay, so then we can think of the information flow through the basal ganglia as being subdivided down multiple pathways. So we have this direct or go pathway. So this is an inhibition of an inhibition, right? So this is removing the brake. So this becomes a go signal. It facilitates action by removing the brake. Next, we have the indirect pathway. And here, instead of having two inhibitory connections, we have 1, 2, 3, right? So that flips the sign. So this becomes a no-go. The net effect of this pathway, as well as this other component of the indirect pathway, is to apply the brake and stop movement. And then third, we have this hyperdirect pathway, which is this fast excitatory signal that says, apply the brake, right? This is, stop everything. So we ramp up the brake and we freeze. Okay, so that's the idea of this framework. And what this sets up then is this competition in the striatum, the input recipient, cortical signaling recipient area of the basal ganglia, between this sort of direct pathway believer population that says, yes, go, acting is the right thing to do, and the skeptic or no-go side, which says, no, we want to hold off, hold that thought. And they're sort of fighting it out to see whether we'll act or not. Okay, so there is evidence for this general framework, and perhaps the strongest to my knowledge are these optogenetic experiments done in the Kreitzer lab. And so here what we can see is if we stimulate the go side, right? So that was removing the brake, right? We stimulate, we remove the brake, the braking cells have shut off, and we should go, right? If we stimulate the indirect side, we apply the brake, we should not go. And so here's a movie from their Nature paper supplement showing this very dramatically, the no-go effect. So we're applying the brake when the light goes on, and the animal just freezes. Right, so that's pretty convincing. All we're doing is stimulating striatal cells in the no-go pathway, and that's enough to freeze the animal. Okay, and this goes on like this through many different trials. So we can see this summarized very clearly in these diagrams from the paper, right? So when we remove the brake, we switch from, these are rodent motion, trails. We switch from this standard gray trail to this very active go trail. And when we apply the brake with the laser, we shut off the movement and we switch from gray to green. Okay, but this is optogenetics, this is a sledgehammer. This is pounding the whole system. Okay, so what about under more natural conditions? And by the way, the basal ganglia isn't just the striatum, right? So what about all of these other areas and all of these other pathways? So this is what we're after, trying to understand the contributions of all of these pathways to this action selection and tuning process. And so there's a major debate in the experimental literature and theoretical literature about which of these two roles is the right way to think about basal ganglia. Is it actually selecting action, right? For example, as stated, in this Nature paper in the bullet font down here at the bottom, the claim is that basal ganglia activity actually starts and stops specific actions. Okay, the other side of the debate is that no, the basal ganglia do not select the reach targets, but what they're doing is controlling the urgency or the vigor with which we perform these movements. Okay, so this is an action tuning role. Now this talk is being recorded, and so I'm not going to go through all these references here, but you're welcome to go back to the recording and sort of pick out what you're interested in. But the bottom line is there's a debate, right? And so as a theorist, this is a great opportunity to jump in with models and analysis and try to make predictions about what's really going on in a principled way. Okay, and my philosophy as a theorist is whenever there's a debate in the experimental literature like this, especially with so many bullet points on each side, there's an easy answer. Okay, the answer is they're both right. Okay, in other words, surely basal ganglia is contributing in some way to action selection, at least under some conditions, but the way that it does so may heavily involve some sort of tuning properties. Okay, it doesn't have to just be selecting the action. So the modeling framework that I'm going to introduce now tries to parse out these different roles. Okay, and I'm going to introduce this thing that we're calling the upward mapping, which did take some inspiration from these papers cited down at the bottom of the slide. I'm going to introduce this in what's called a two-alternative forced choice task. Okay, so think of like a monkey or a person for that matter, looking at moving dots and having to saccade to one target or another, depending on whether the dots are predominantly going left or right. You can think of a rodent in a T-maze, right, having to choose one of two arms to get a reward. Or you can think of the many different situations we encounter where we're going to make a decision and we're choosing between a finite, in this case, two dimensional set of options. Okay, so in this setup, there's a really convenient mathematical model to represent what's happening in an algorithmic way in when we make decisions. Okay, and this is the famous drift diffusion model, which has been generalized to more than two decisions, but as I said, I'm focusing on two choices here. Okay, so in the drift diffusion model, we have a random walk in a bounded domain with a drift component. Okay, so the drift rate biases us towards heading towards one boundary or the other. And so we have a parameter, which is the strength of that drift, that's V. So you can see when you tune V, you're going to change the reaction time distribution. Okay, and the idea is when you feel like the evidence is stronger for that option, you should tune up the drift rate. Okay, we also have a boundary separation, so some sort of degree of skepticism. So we can tune the speed accuracy trade-off by tuning where the boundary height, or how separated the boundaries are. Okay, we can have a starting bias, which has a degree of freedom Z. In our setup, we happen to use Z times A, but so Z is between 0 and 1. But in any case, this says how biased we are towards one decision or the other to begin with. And then a fourth parameter that we use is the onset time, TR or T, which is just how long we wait before we start taking into account evidence in making the decision. Okay, so this allows us to really clearly put all of the aspects of decision making in these intuitive terms, right? Like how strongly we accumulate evidence. And what we want to do is connect this to the actual neuronal activity, right? Figure out which neuronal components are tuning which aspects of the decision making or the action selection. Okay, so to do that in our simulations, we set up two different action channels. So basically two copies of all of these different populations in the basal ganglia. Okay, again, don't worry about what the abbreviations stand for. Whoops. And so one of these action channels is related to choice A, the other is related to choice B. Okay, and so what we do is we have a random process in the cortex that is driving upward the level of activity in the cortical populations. Okay, and eventually this filters through the basal ganglia till it gets down to the thalamus. Okay, and there's a thalamic population, and we say that a decision is made when that thalamic signal, whether it's due to cortical input or basal ganglia factors, when that thalamic signal hits a high enough level, then the action signal is free to progress. Okay, so what happens is, as the action ramps up, or sorry, the evidence or activity ramps up in cortex, it's also ramping up in the striatum. And this is the go side, right? So this is trying to turn off the brake. Down here is the brake, right? It's what's inhibiting the thalamus. As this ramps up, it's trying to turn off the brake. On the other side, we have the indirect pathway, which is trying to keep the brake on. Okay, and for a while, the competition is pretty even, right? The brake stays at a constant firing rate, so a constant level. But eventually the balance is broken. Okay, and this is because, in a nutshell, firing rates can only go so low. And so in the place in the circuit where firing rates are going down instead of up, you sort of hit a saturation point, which eventually breaks the balance. So when the balance is broken, the thalamus on one side hits a threshold and a decision is made. Okay, so if we do that over and over and over again, we're going to get a whole distribution of reaction times. And we're also going to get a fraction of A choices and a fraction of B choices. So even though that's generated from simulations, we can use that as data, right, to fit with the drift diffusion model. So the collection of this output gets fit by the drift diffusion model. Okay, and we can do that in various ways and come up with the best fit. And we can interpret this as a mapping from the neuronal parameters, right, the weights of the synapses in this network, to the parameters of the drift diffusion model. Okay, so let me show you what this looks like. So the logic, again, is think of two nested loops. Okay, so in the outer loop, we are going to choose a set of basal ganglia weights. So all of these different synaptic weights in our network. Okay, for that set of weights, we are going to run many, many trials of simulating this network, basically doing this simulation that I showed you. From all of those trials, we are going to collect reaction time distributions, mean firing rates, and our percentages of A and B choices. Right? So that looks like this over all of the different trials. Here we've done 300 trials. And then we fit that behavioral data with the drift diffusion model, which is going to give us a set of parameters for that set of weights. And then we iterate the outer loop again. So we do this over and over again, okay, until we get a whole big collection of data, which might look like this. So I'm not showing you the weights. We have, in this case, 300 different sets of basal ganglia weights chosen using Latin hypercube sampling, which I can talk about more in the questions if you want. This is what the mean reaction times look like from those 300 trials. This is the percentage of A's, which we expect to be on average 50%, because we didn't introduce any bias towards one choice or the other. And these are the distributions of the 4 different drift diffusion parameters that we got out from our fitting process. Okay. Right, there's lots of details here, which again, we can talk about in the questions. So what do we do now that we have all of this data, right? So one thing we could do is cluster this, right? There's sort of looks like maybe there's a couple of different clusters. What we decided to do that was most informative was something called canonical correlation analysis. Okay, so if we think of this, as I described earlier, as this mapping from the weights in the neural network to the parameters of the drift diffusion model, okay, we want to use canonical correlation analysis to pick out the projections, right, which I'm calling WA and WB down here, not because they're the weights in the network, please ignore that notation, but there are projections, okay? We want to pick out the projections such that when we project our paired observations under these projections, we end up with the strongest correlation, okay, in the range, okay, among all of these vectors. And in fact, if you, right, you can choose a size of your projection. Here it was chosen to be 2-dimensional, but by choosing different sizes of projections, you can find the one that gives you the best ability to predict the data from these projected vectors, okay? So if you want more information on this, I took this diagram from a really nice blog by a mathematician named Gregory Gunderson, and he explains CCA very well. What we did was to decompose our mapping, our upward mapping, into 2 components. Okay, so we go from weights to rates in the basal ganglia itself. So these are both relatively high dimensional. And then we go from rates to DDM parameters. So higher dimensional to just 4-dimensional. And it turned out this was the key to getting information, because what really matters for the output of the basal ganglia is the rates. Okay, different combinations of weights can give you the same rates, so that can obscure some relationships, but we found nice relationships between rates and DDM parameters. Okay, so methodologically, hopefully this is useful for other situations that you might be interested in, where you want to link neuronal activity to some sort of behavioral features. And now I'm going to show you what we got when we did this analysis on our basal ganglia model. Okay, so we found that 3-dimensional projections were the most useful for this data set. Okay, and these are the loadings for our 3 dimensions. So over here on the left, we have the firing rates. Over here on the right, we have the DDM parameters. Okay, so R and P. The loadings show us the coefficients of the vectors in the projected vectors that we get, okay, for each of these important components. Okay, and here in purple means negative coefficients, green means positive. Okay, what we have here for the firing rates, for reasons that I can talk about, we didn't look at the A GPI, B GPI, ASTN, B STN, and so on. What we did was we looked at the sum in each basal ganglia area across the 2 channels and the difference between the 2 channels. And if you think about it, that is really the most informative aspect of the firing rates. And over here, again, we have the DDM parameters. Okay, so to understand, unpack all of this information, we go back and we draw diagrams where we take these loadings, right, and we color code the corresponding basal ganglia populations according to these loadings, and we color code the drift diffusion parameters according to these loadings. So for example, for the loading that was loading 0 on the previous slide, now I'm calling it 1, okay, we color code these basal ganglia areas, and this gives us what we call the responsiveness control ensemble. So these are collections of changes, in the basal ganglia in terms of the sums of firing rates across the 2 channels that most effectively tune some aspect of the DDM, okay, needed for the fitting. My mouse is very sensitive. So here, for example, the DDM, the parameter that shows up most heavily in this first component is the onset of evidence accumulation. So that's why we call this the responsiveness ensemble, right? How responsive are we? How long are we going to wait before we start collecting information? And this is purple, so it's negative. Okay, so things that are green over here, when these increase, so when the overall GO pathway activity increases, that's going to correspond to an earlier onset time. So this is very consistent with the classical notion of the GO pathway, when the overall activity in the output of basal ganglia and the DSPN cells for the GO pathway gets stronger, that's going to say we're going to start accumulating information earlier. Okay, and I don't want to belabor the details because, again, I know you're not all a basal ganglia audience, but these are in a paper which we've revised and is now under second review. So I think it's on the archive and it hopefully will be out before too long. But in any case, to quickly mention, our second control ensemble that we find is for something we call pliancy. Okay, so that's not necessarily an everyday vocabulary word, but what pliancy refers to is our sort of willingness to be convinced, right? And so in this component, it's the boundary separation or boundary height that's loading relatively strongly. So how much evidence do we need to be convinced? And here we can see in green the populations whose overall activity is contributing strongly to pushing up the boundary height, and in purple, the populations that are trying to pull down the boundary height. And so we can see that the indirect pathway becomes strongly involved in that, right? And so when we drive the indirect pathway more strongly, again, like increasing the brake, it turns out the way this is happening produces a reaction time distribution that's consistent with an increased boundary height. And then our third and final important component is what we call the choice component. Okay, and this is the one that impacts the drift rate most strongly. So how oriented towards one choice or the other are we? And this is the one where the difference in activity between the two channels becomes most important. Okay, so large interchannel differences in certain areas correlate with faster evidence accumulation. So that would be the green ones, and the purple would be slower evidence accumulation. And so here we can see not only the competition between the channels, but the competition between the GO and no-go pathways. Okay, so again, like the details of this, I think require thought and are difficult to absorb probably in this setting, but this gives you the flavor of the types of information we can get out of this analysis. Okay, and so to summarize this first part of the talk, what I've shown you from the basal ganglia perspective is that we've identified these control ensembles that modulate specific aspects, both of the action selection process, right? But also the tuning of how we select actions, right? How long we need to wait, how willing we are to move forward, how urgent or responsive, we are to signals that come in. So this is really both action selection and tuning. Okay, and so our prediction, this is the learning in the basal ganglia, right? Our prediction is that learning, which should be aiming to adjust decision policies, can do so most effectively if it does this in a coordinated way across a control ensemble. So that's going to give us various predictions for how activity and weights in the basal ganglia may change with learning. And that's what we're looking into now. Okay, and then methodologically, again, this upward mapping approach allows us to link neural activity with behavior. Okay, so the collaborators on this part of the work, the initial collaborators, the collaborators on the published work are here. Tim Versteinen and various lab members from Carnegie Mellon, and Kati Vick from Mallorca. And we have a bigger team now working together on some of these additional aspects about the plasticity, implementing plasticity in the circuit and connecting this with experimental data. Okay, so questions are coming at the end of the talk, so I'm going to keep going onto the second part of the talk. Okay, and this is where, this is like time for the confession, right? So we all have these secrets, and they aren't very secret, right? When you build a model like this, you're making a huge number of assumptions. Just to name a couple, right? We're using single compartment neurons. They are spiking neurons, but they're single compartment. But one that is a little more taken for granted that, we don't really think about a whole lot is I have all of these pathways with balls and triangles, and they're supposed to be inhibitory and excitatory. And that's the dogma, and that's how we treat them in our models, and full stop. But in fact, they're biology, right? They're biological, and there are ions and membrane channels involved, and they might not always act that way. Okay, and so there's a paper sort of lurking in the background from 10 years ago, where they did optogenetic experiments in slices. So not so different from the Nature paper I told you about, but they completely focused on what happens when you stimulate either the go or the no-go side of the striatum, and you record in the braking area, right, the output of basal ganglia. And so remember, we said, okay, when you activate the go pathway, you're supposed to be removing the brake, right? You're supposed to be doing exactly what's shown here. This is the brake population, and you can see the brake is getting shut off. Okay, but in a bunch of their experiments, that didn't happen. In a bunch of those experiments, the brake got stronger. You can see that on this side. The question mark exclamation is mine, but I'm sure they were thinking that. They just couldn't put it in the paper. Okay, likewise, in the indirect pathway, that's supposed to be applying the brake. And you can see that it worked some of the time, right? Stimulate that side, the brake gets stronger. But some of the time it didn't. It's really striking in these rasters, and you can see in the summary data, there are plenty of cases when the population did the wrong thing. So that looks like trouble, right? So what's going on here is something that we decided to model. Okay, and so these results are published, and they're in a 2020 eLife paper. And what we did was we modeled these output SNR neurons, the brake, including the contribution of chloride dynamics to their responses. And then we considered the functional consequences of this diversity of responses in terms of what the basal ganglia output is actually doing. Okay, so here's a schematic of the model. We had to use 2 compartments because these SNR neurons are getting the direct pathway input, so that's here, out in the dendrites, and they get the indirect pathway on this input on the soma. Okay, so we have those 2 compartments, and in addition to the usual Hodgkin-Huxley stuff, we have the chloride potassium exchanging pumps, okay, that bring chloride out of the cells. We have chloride influx through these GABAergic pathways, okay, and we're tracking the chloride concentration in each of these compartments. So we have a copy of this equation in each of these compartments. Okay, and the chloride flux here, the change in the chloride comes, as I said, from the pumps and from the GABA current. And so we have to compute the fraction of the GABA current that is actually chloride ions. Okay, so that's here. And when you take into account all of the reversal potentials, okay, you get the appropriate dynamics for chloride. Okay, and what we find when we do this is that our model does reproduce these diverse responses to these 2 types of inputs. Okay, moreover, we get some interesting implications of how the cis circuit might actually use the chloride load to change action selection and SNR output cell firing, including some possible implications for Parkinson's disease. So in my remaining few minutes, I'm going to talk about that. Okay, so here is just showing you that we did more experiments. The Gittes lab at Carnegie Mellon did more experiments and showed that they got the diversity of responses seen in the earlier paper. So there are examples in this figure and quantification of how frequent the different responses were. Okay, and by tuning our 2-compartment model to different conductances of the pump and the tonic chloride load. So we assume that there's an ongoing chloride load that was already present from the ongoing activity of GPE. By picking out different places in this space, we get all the different responses seen experimentally in the model. And you can see that the GABA reversal potential can gradually change over the course of the stimulation period. Okay, and that can lead to a partial inhibition or a biphasic response like this, right? And so this is what happens when you stimulate the striatum, so the GO pathway. So that's going into a smaller compartment. The dendritic compartment we treat as smaller, so it can have this more drastic effect that can lead to more biphasic, switches from inhibition to excitation. Okay, so functionally, what does this do? What do we think this might do? Okay, so one of the ideas here is the GPE is generally firing at a high rate. And whatever that rate is going to set this chloride load, right? So it's going to set sort of where we are on this axis, which, as you can see from the colors, sets the GABA reversal potential. Okay, so we can move horizontally depending how active the GPE is. Okay, and then if we think of the role of the GO pathway as trying to pause the brake to allow action to proceed, okay, how easy or difficult that's going to be is going to depend on how effective this inhibition is, which depends on what this chloride load is that the GPE has imposed. So this is easy to demonstrate if, depending on the firing rate threshold we use to define stopping the brake. Okay, it takes different amounts of time for the SNR firing to lower to that level as the striatal activity ramps up with evidence. Okay, and if we fix some chloride load, say, and fix some threshold, say the red one, and vary the chloride load, as the chloride load goes up, the reversal potential for GABA goes up. It takes longer and longer to pause. So that gives us a functional mechanism to slow down our actions, right? So to reduce the vigor of our action selection. Okay. A second implication is within the SNR network itself. Okay, so SNR neurons are coupled to each other with inhibition, or GABA anyway, but it's sparse. Okay, so again, if the GPE is setting the chloride load and therefore the GABA reversal potential, then the effect of these SNR GABAergic collaterals will depend on where that reversal potential is set. So for example, if it's nice and hyperpolarized, then when a GABAergic input comes in, it will delay a spike of an SNR neuron from where it would have been to a later time. Okay, but if this is depolarized, it can actually advance the spike time. Okay, and this is similar to results by Boris Gutkin in the past. Okay, just working out these PRCs for different levels of E-GABA. So the PRCs show us, depending on what phase a stimulus comes in and the level of E-GABA, to when will this stimulus advance the next spike or delay the next spike. Okay, and SNR neurons are oscillators, so they always have a next spike. Okay, so in these small circuits then, if we go down to the smallest toy circuits we can play with, we can look at the PRC for different levels of E-GABA. So 3 different ones are shown here. And in a feed-forward circuit with the same baseline rates for the 2 cells, we can see that for hyperpolarized E-GABA, we don't have a fixed point. We have an unstable fixed point, sorry, where this crosses 0. Whereas when we raise E-GABA, we can stabilize a fixed phase relationship between these 2 neurons. Okay. In a bidirectional circuit, it's much less interesting. We basically always have an anti-phase stable fixed point. Okay, so if we look then at where this fixed point is and whether it's stable or unstable, we run simulations of these simple SNR circuits under these different levels of E-GABA, different chloride loads from GPE, and we get these different phases of spike time relationships between the 2 neurons, which fit very well with the predictions from the fixed points. And we can see when the fixed point is unstable, we actually get oscillations, phase slipping oscillations, where the second neuron falls behind the phase of the first one due to the inhibitory action of GABA, and its firing rate slows down. Okay, but tuning up E-GABA, we no longer have those oscillations because we stabilize a phase relationship. Okay, depending on how we tune the presynaptic firing rate relative to the postsynaptic firing rate, in fact, we can either get phase slipping oscillations on low E-GABAs, or we can get phase advancing oscillations at high E-GABAs. All right, so why would this matter? Well, one more thing before that. we run a simulation of a full SNR network. So now 100 neurons, okay, not quite full at a neural scale, but much larger than 2. We use data from the Wilson lab to randomly select the number of GABAergic connections between the different SNR neurons. And we look at the power in the 0.5 to 4 hertz frequency range, which you'll notice, right? The frequencies, I forgot to point them out, but they're in that range for these types of oscillations we see. And we see that for low E-GABA or for high E-GABA, we find these oscillations in various cells in the circuit. And for intermediate E-GABA, they're gone. Okay, so it's easy to imagine that the normal regime is this one, and a Parkinsonian regime could be one of these, where we start to get these pathological oscillations. Okay, and David, I'm planning on talking for maybe 3 minutes more. I hope that's okay. So in these last few minutes, what I'm going to say is related to these oscillations. Okay, because oscillations in Parkinson's disease are famous, but they are not 0.5 to 4 hertz oscillations like these are. They are beta band oscillations, right? They get all the headlines. Those are 7 to 35 hertz. Okay, well, it turns out in the mouse, there aren't any beta oscillations. Okay, in the mouse, and this is data from a paper that we did in collaboration with the Gittes lab. In the mouse, when you deplete dopamine, bilaterally, unilaterally, or with a channel blocker, you see delta oscillations that are not there under control conditions. And this is in the SNR still. You also see it in other areas. And you don't see any beta oscillations. Okay. Now, interestingly, you do see these low frequency oscillations in humans, okay, with Parkinson's, or at least dopamine depletion. Okay, now, often when there are low frequency oscillations, people attribute them to the cortex, filtering to the rest of the brain. But Tim actually aspirated the M1, the basal ganglia projecting cortex out of his mice, and he still got the oscillations under dopamine depletion. So they're not cortically generated. Okay, maybe they're an epiphenomenon. Maybe they're actually just a fluctuation, right? How do we know they're real? Okay, and the answer is because we use this phase shift test. Okay, so in very brief, the idea of the phase shift is for each frequency that we're interested in, that we identify in our signal, right? We're identifying this in different time segments of data. And so we imagine we have a perfect oscillator at that frequency that started at the beginning of the simulation. Okay, and when it gets to this time segment that we're interested in, we call whatever phase it's at phase 0. And we realign the phase of the time series relative to that phase 0. And if it's a true oscillation in the time series, then it should maintain a reliable phase relative to the perfect oscillator across different time segments. So we compute the phase shift between time segments, and you can see in the real oscillation in this cartoon, it stays low. And in some other frequency band where there isn't a real oscillation, the phase shift will jump around. Okay, so we would rule that out. And so we get in the data a bunch of true positives where the phase shift is low and the power is high, and a bunch of false positives where the power is high, but the phase shift is not low. So we don't call those oscillations. Okay, so final slide. What we predict, to tie this back to our model, is that whether these oscillations are present or not should relate to E-GABA in the circuit. And so the idea would be, we claim that stimulating the GPE sets the chloride load, which tunes E-GABA. So we predict that the chloride load is normally low under dopamine depletion because GPE activity is down. So if we stimulate GPE, we will raise E-GABA, and that should move us from this oscillating regime to this non-oscillating regime. And in brief, in 5 animals at least, that's what we found. Okay, so stimulating the GPE killed the oscillations at low frequency in the SNR without changing the firing rate. All right, so here are my conclusions from the latter parts of the talk. What I'll just let you read them. And I want to emphasize that we should all be thinking about chloride dynamics in circuits with GABAergic synapses with lots of inhibitory activity. And then I want to thank my collaborators. So Ryan Phillips, who's now in Seattle, members of the GITIS lab from Carnegie Mellon, including Asier Aristieta, and then my current postdoc, John Parker, who are working with us to try to push these results further. So thanks a lot for your attention.